The term biotechnology refers to the use of living organisms to modify human health and the environment. Throughout history, we have learned a great deal about the different organisms that our ancestors used so effectively. Increased technology has helped biologists study the organism's cell structure and function, and they have found ways to use these organisms even more effectively than our ancestors. However, biologists must have a basic understanding of cells and their many functions before manipulating their gene expression for our benefit. Understanding what makes up a cell and how the cell works is fundamental to all of the biological sciences. A cell is the smallest unit of life and the building block of all living organisms. Appreciating the similarities and differences between cell types within and among organisms is particularly important to the fields of cell biology and molecular biology. Molecular biology can be confusing because it is a subject that focuses on things so small you cannot even see them. So let's begin with big organisms and work down to the molecular level. Organisms are made up of organ systems, such as the digestive system, pictured here. Each of these organ systems are comprised of individual organs. As shown in the diagram, organs such as the liver, the stomach, and the small and large intestines are part of the digestive system. All of these organs are made up of tissues. Tissues are a group of similar cells that perform the same functions, such as the epithelial tissue and the smooth muscle tissue pictured here. Tissues are made up of cells, and a cell is the smallest unit of life. Finally, cells contain organelles and molecules such as DNA. Let's examine a cell more closely. There are two general classifications of cells, prokaryotic cells and eukaryotic cells. Prokaryotes are single-celled organisms and include bacteria and some algae. On the other hand, eukaryotic cells make up more complex organisms like plants and animals. Prokaryotic cells lack a membrane-bound nucleus, so the DNA, which is usually a single circular molecule, floats in the cytoplasm of the cell.
The object which you see is a model of an animal cell. Animal cells are one of the most basic types of eukaryotic cells. Animal cells are the foundation of all animals, which are made up of multiple cells and which have a nucleus and membrane-bound organelles. The main parts of the animal cell are the mitochondria, microfilaments, lysosome, Golgi apparatus, smooth ER, rough ER, microtubules, cell membrane, ribosomes, and nucleus.
The bilayer surrounds the cell, protecting it, and functions as a gate that allows molecules into and out of the cell. In our city, the cell membrane would be like a large fence surrounding the city that only allows certain molecules to go in and out of the cell.
This is an example of a nucleus. The nucleus controls most of the functions that go on within a cell. The nucleus also contains a cell's genetic information. This is an example of mitochondria. Mitochondria are small organelles that transfer energy from organic molecules to ATP. ATP is the main type of power which runs a cell, making the mitochondria very important. This is an example of a ribosome. Ribosomes are somewhat spherical organelles that serve the purpose of building protein. Also, ribosomes are created by the cytoplasm. The Golgi apparatus is a system of membranous sacs. The purpose of the Golgi apparatus is to transport vesicles and the things which they carry.
The endoplasmic reticulum, it forms an interconnected network of tubules and vesicles and functions to transport proteins throughout the cell. So those protein-containing vesicles would be like the trucks on the highways of our city. Many of the protein-producing ribosomes are located on the endoplasmic reticulum.
This is an example of a lysosome. Lysosomes are vesicles created by the Golgi apparatus which contain digestive enzymes.
There are actually two types of eukaryotic cells, animal cells and plant cells. Both types of cells are very similar, but there are three distinct differences. First, plant cells have a cell wall surrounding the plasma cell membrane. Animal cells just have a cell membrane. Secondly, there is often just one large central vacuole found in plant cells, whereas animal cells have a few small ones. The vacuole stores molecules needed by a cell and also isolates material that may be harmful to the cell. The final difference is plant cells have an additional organelle called a chloroplast. The chloroplast is the site of photosynthesis in a plant cell. Photosynthesis is the process of capturing sunlight and synthesizing glucose and oxygen from carbon dioxide and water. Photosynthesis is one of the most important biochemical pathways since nearly all life on Earth depends on photosynthesis as a source of energy. Animals metabolize the glucose from a plant source to produce ATP, the energy currency of the cell. Recall that all organisms need energy to do cellular processes.